Hello teacher, I tutor about Germline mutation in predisposition, hence in pediatric cancer. I study in Catholic University, Nursing School St. Vincent of Paul, Technical English. My name is Nadia Barriga Mendez. I'm teacher, Master John Moscoso. Definition: RASH. Genes encoding similar proteins involved in G-tyrosine growth and the differentiation signals from tyrosine kinase receptor to core cells. The NRAS gene is located on the short arm of chromosome 1 at molecular level. The level being case of collateral cancer associated with mutation in exon 2, 3, and 4 of this gene.
My dad was the first generation of this disease in our family, and it is cancer of the retina.
Cancer predisposition syndromes in general are a group of conditions where a person, because of changes in their DNA, is at risk for cancer.
Von Hippel-Lindau, it's a genetic disorder. My grandma had it, and then my dad got it, and then I have it, and my two children have it.
We decided to try and create a clinic where these children who have known syndromes could be watched, and we could build a research repository to study their conditions in more genetic detail.
The Cancer Predisposition Clinic is a newer idea. What we do is to identify individuals who are at risk, often because there is a cancer history in their family.
When I started having children, we knew the chances of it. Within 2 weeks of age, he was diagnosed.
Depending on which cancers tend to go with a particular syndrome, we put together a set of screening protocols that identify early the tumors that person may be at risk for.
Cancer in children is not adult cancer in a small body. It is a unique set of diseases that have a unique set of genetic causes and requires a unique set of treatments.
We do genetic tests to look at the spelling of genes, to look for the changes that tell us that person is at risk of cancer.
When we came, they did a comprehensive exam of both boys. They looked at everything. They were looking at those imperfections and seeing if they were linked together to see if other children with retinoblastoma may also have, a thin lip, and that's a precursor to something else.
The goal of these tests, however, is to be able to identify tumors when they're small, early on, when they have much more chance of being able to be easily surgically rectified. To be able to bring together the level of expertise that you have, your genetics docs, you have the oncologists, you have the specialists in terms of the other organ systems involved, I truly think is the best way for patients to be taken care of.
We're lucky that we have a place to go that can find things early, so we can catch things before it's advanced.
In 2000, the cost of sequencing one person's entire genome was over $1 billion, and today we can do that in less than a week for about $1,000. So while it seems a bit out of reach today to be doing comprehensive genomic analysis on every child that comes through the door, those days are not far off. And as we recognize that more and more children's cancers probably stem from the genetic profile that they're born with, we want to have all of the samples and tools available in place already to rapidly proceed through the research.
It's a good feeling to know that if something is wrong or going to go wrong, they're there to catch it.
The goal is definitely to both screen for these cancers so that we can detect them early, and also to find new information in the patients that do get cancer that tell us what was different about them. And I think that's ultimately going to be what's important. I mean, it's wonderful if we can detect a cancer early. It's even better if we can do something to make sure that cancer doesn't happen.
It's exciting to be a part of it, to be able to give our information so that it will help other people.
Children's is where I wanted to be if anything ever did occur.
Because our samples continue to evolve and our collection grows, I really believe this is going to become a model for how multidisciplinary pediatric care can go forward through the genetics prism.
Application in medicine. The researchers analyzed the genomes of 1120 children and adolescents with cancer and looked for genetic variation in 565, hence associated with cancer with special spines of 60 genes associated with autosomal dominant cancer predisposition syndromes.
Hi, I'm Rob Grant. Today I'll be discussing our study, Prevalence of Mutations in Cancer Predisposition Genes in Patients with Pancreatic Cancer. Our lab is particularly interested in pancreatic cancers that are caused by inherited mutations in the BRCA1 and BRCA2 genes. These genes are famous for causing breast and ovarian cancer, but also increase the risk of pancreatic cancer by up to 7 times that of the general population. Identifying patients with BRCA mutations can have important benefits. Relatives should undergo genetic screening, and carriers of mutations may benefit from interventions like bilateral mastectomies and salpingo-oophorectomies. Also, emerging evidence suggests that pancreatic cancers caused by BRCA mutations have a distinct genomic landscape, prognosis, and response to therapy. Clinical trials are currently underway investigating agents that specifically exploit the defective DNA repair pathways in BRCA-deficient pancreatic cancers. We don't know how much pancreatic cancer is caused by mutations in the BRCA genes, because studies to date have focused on cohorts selected for family history or ethnicity rather than population-based cohorts. Other genetic syndromes significantly increase the risk of pancreatic cancer. These include Lynch syndrome, familial atypical multiple mole melanoma syndrome, familial adenomatous polyposis, hereditary pancreatitis, Peutz-Jeghers syndrome, and Li-Fraumeni syndrome. The benefits of identifying carriers of mutations that cause these syndromes is similar to that of the BRCA genes. The goal of our study was to define how much pancreatic cancer is caused by mutations in known predisposition genes. We sequenced 13 genes that cause the syndromes I just described in a population-based cohort of pancreatic cancer patients. We wanted to detect at least one mutation in any gene mutated in over 1% of pancreatic cancer. To achieve this with adequate power, we selected 290 of 708 probands with pancreatic cancer from the Ontario Pancreas Cancer Study. The OPCS is a population-based cohort that recruits all consenting participants with a pathological diagnosis of pancreatic cancer in the province of Ontario, Canada. To maximize the precision of our estimate of the BRCA mutation rate, we randomly selected patients from 3 strata based on family history of BRCA-associated cancers. Lymphocyte DNA from all 290 probands underwent targeted next-generation sequencing. Our bioinformatic pipeline detected genetic variants in the pancreatic cancer predisposition genes. Rare, non-synonymous variants were classified as either pathogenic, of unknown significance, or benign, based on their impact on the protein and publicly available databases. Each position in the targeted genes was covered by an average of 354 sequencing reads. Most rare non-synonymous variants were of unknown significance. On average, each proband carried 1.2 variants of unknown significance. Longer genes had more variants of unknown significance, supporting a more benign role for these variants. We detected 11 pathogenic variants that were then confirmed with Sanger sequencing. Although most had a history of cancer, none met the Amsterdam 2 criteria for Lynch syndrome. Only one patient had a first-degree relative with pancreatic cancer. The 2 available tumors from carriers of mutations in the mismatch repair genes were immunodeficient for their respective protein. 3.8% of probands carried a pathogenic mutation after accounting for the stratified sampling design. This corresponds to about 1 in 26 pancreatic cancer patients. 2% of probands had mutations in genes associated with breast cancer, and 1.4% in genes associated with colon cancer. The only characteristics significantly associated with mutation status were family and personal history of breast or colon cancer. The carrier rate in probands with colon or breast cancer in their personal history or in a first-degree relative was over 10%. Our study supports the use of multiple gene panels in pancreatic cancer, considering the breadth of mutations detected and and the wide range of personal and family history of cancer in the mutation carriers. Our study also raises questions. Which pancreatic cancer patients should be referred for genetic screening? Do mutation carriers with atypical family histories of cancer face the same risk of cancer? Ongoing international collaborative sequencing efforts may provide answers to these challenging questions.
Pros and cons. The study of the tumors developed by carriers revelates that the mutation identified in hereditary colon cancer found one does not behave a classical suppressor hence, but that alteration of a single allele will sufficient to trigger a failure in DNA repair which ultimately causes the development of a tumor. Genetic factor affecting 4 to 10% of pediatric cancer. The molecular biology techniques are most identified, hence involved in familial cancer. The diagnosis of these syndromes allows genetic study of patients and their families. Genetic counseling should provide useful prognostic information data, evolutionary monitoring, early detection, and even prophylactic treatment of associated cancer.
Cancer is a disease in which cells divide uncontrollably. Over the past 30 years, detailed studies have shown that genes, such as tumor suppressor genes, are often destroyed by DNA mutations, allowing cancer cells to grow. In recent studies of certain breast cancers, it has been observed that parts of the DNA that control tumor suppressor genes are frequent targets of cancer mutations. First, let's explore a normal cell, which is not dividing. Inside the nucleus of the cell, transcription factors are constantly scanning the DNA for their binding sites. The transcription factor moves along the double helix until it encounters a place where the nucleotide pattern is in the exact sequence for it to stick. Once the exact sequence is found, the transcription factor changes shape and binds tightly to the DNA. This binding allows the important tumor suppressor gene to be expressed. Many proteins then stick to the transcription factor and form a protein machine to make RNA for the tumor suppressor gene. This production prevents the cell from dividing. Now let's explore a cancer cell. Like all cancer cells, this cell divides when it is not supposed to. Inside the nucleus, the transcription factor searches the DNA for its binding site. Different types of cancer have unique mutations. In this specific type of breast cancer, the binding site has been mutated. The altered nucleotide pattern is not recognized by the transcription factor. It passes by without sticking. Without the transcription factor binding, the gene stays inactive and the critical tumor suppressor gene is not expressed. The cancer cells continue to divide without control. Over time, more mutations accumulate, causing the cancer to grow faster and faster.
Conclusion. New therapeutic agent acting on the receptor of epidermal growth factor, such as cetuximab and panitumumab, have proven effective against colorectal cancer. New evidence suggests that the presence of an NRAS mutation is associated with a poor response to this stage of treatment. Patients with colorectal cancer will have rumors they carry the native NRAS, hence without mutation, will benefit from egg-free antagonist-based therapies.
The tumor suppressor protein retinoblastoma is found on chromosome 13. Retinoblastoma's structure has two domains, A and B, that form a pocket domain permitting binding of E2F. You can think of him as your friendly police officer. E2F is a transcription factor that promotes cell cycle progression by producing proteins, including cyclin-dependent kinases 4 and 6 and cyclin D. Extracellular signals, such as stress, can also increase cyclin D levels. You can think of him as your criminal ringleader with his accomplices. Cells go through a number of cell cycle stages to proliferate, including GAP1 phase, synthesis, GAP2 phase, and mitosis. Our friendly officer RB and criminal ringleader collide in G1. Hypophosphorylation of officer RB results in the arrest of ringleader E2F, locking him away and preventing his nefarious acts of initiating proliferation. As G phase progresses, RB is increasingly phosphorylated by increasing cyclin D CDK4/6 complexes. Phosphorylation inhibits E2F binding, releasing the E2F. This could also be seen as E2F's accomplices coming to the rescue and bribing Officer RB to release their ringleader. Now free to get back to his usual antics, E2F causes proliferation by entering the nucleus and promoting transcription of proteins required for S-phase progression. When Officer RB is mutated, he's no longer able to bind E2F. These may be inactivating mutations or promoter hypermethylation, resulting in gene silencing or increased sequestration, causing E2F to allow premature cell cycle progression and excessive proliferation. Such unregulated proliferation, if not stopped, can lead to tumorigenesis. Mutations in the RB gene are known to cause retinoblastoma cancer, which develops in the retina of the eye and is typically found in children. Other cancers linked to the RB gene include bladder, lung, breast and bone cancers. Thank you for listening.
Thank you.